Okay, this is part two of chapter three, which mostly deals with the ocean. So we're giving kind of equal time to the land and the ocean, although we all know the ocean deserves more because it covers 71% of the Earth's surface and contains a rich diversity of life. Marine biological zones are categorized by their location relative to shorelines and the ocean depth. And marine organisms are influenced by temperature, light, water depth, bottom substrate, and interactions with others. So those are sort of like the main things we think about when we're, trying to figure out how critters make a living or how they interact with one another. So this is breaking the ocean down at its distance from shore, from near shore, which it's kind of funny. Near shore isn't really that well defined. I mean, there's arguments in the oceanographic realm about what that would mean. I would kind of define this a little bit differently, but near shore really could mean the majority of the Salish Sea. It's a relatively near shore environment. Here we've got a water depth of 200 meters. There are places we get 600 feet or more in the Salish Sea, particularly if you include the straits. But you can see shore wherever you're at, right? So you're relatively near shore. And then what they're calling shallow ocean here, I might think of that as more the continental shelf, which can extend a number of miles offshore. And then the pelagic is being the more deep ocean that's not, waves are not coming in contact with feeling the pressure of the bottom of the ocean. And you're more of a, blue water system. The photic zone is defined, and now we're going depth. The photic zone is defined as the parcel of the ocean that light penetrates. It's listed here as 0.5 to 200 meters because that depends on the water clarity. For example, one way you can measure water clarity is with what's called a Secchi disk. It's a really simple tool. It's a circle, like a frisbee, with alternating black and white triangles. So you can imagine a frisbee, tape off two triangles and spray paint the other two black. Lower that down, and when you can no longer make out the differentiation between the black and the white, you mark that depth. It's a way of measuring water clarity. In Bellingham Bay, you can have Secchi depths of really just a few centimeters because there's so much sediment coming out of the Nooksack River. You go out in the open ocean, and maybe you can see a few hundred feet down. And so it's like this interesting tool that's been used forever, and it's remarkably good. And then the benthic or benthos just being the bottom, whether you're in the intertidal or in the abyssal zone or the Marianas Trench, the bottom is still called the benthic. So here they say near shore zones are influenced by local climate, tides, waves, and influx of fresh water and terrestrial sediments. So that definition of the near shore is having some interaction with the benthos. Now waves can feel the bottom, or waves are altered by the bottom, even though the bottom's much farther down than they are. So we often think of waves breaking, like a wave breaking in a few feet of water. But the currents are impacted by the bottom, so a current could be impacted by the bottom, even if it's 1 or 2, 3, 400 feet deep. And so that's one kind of measure of what's near shore are the surface currents feeling the tension or interacting with the benthos, as well as this idea of some form of terrestrial input, whether it be nutrients or fresh water, etc. And tides is the bathtub moving up and down. Here we have what's called mixed semi-diurnal tides. So here we've got, it's kind of weird, we have 2 high tides and 2 low tides a day. So we'll refer to that as a higher high and a lower high, and a lower low tide and a higher low tide. So we actually have multiple tides and multiple types of tides a day. Other areas that's not true. They'll have one high tide, and then 12 hours later they'll have a low tide. Where here we're about every 6 hours or so we change. It's like 6.5. It's a bit different. And estuaries are where the rivers meet the oceans, and you can really define estuaries as some area with freshwater influence. Technically, all of Puget Sound is an estuary. Technically, all of Puget Sound has a freshwater influence, and I mentioned that in one of the earlier lectures, that the salinity in Puget Sound is about 28 compared to the open ocean, which is 32, 34-ish. And they're typically really productive areas because they're a combination of freshwater and marine, and a lot of nutrients coming out of the freshwater entering the marine environment. Typically areas that have softer sediment. So the true part of the estuary can have a lot of both like salt marsh grass and marine plants and algae, and fix a lot of carbon, have a lot of primary productivity. Additionally, estuaries are good rearing areas for the juvenile stages of fish and I say shellfish, which is like not a taxonomic term, but crabs, etc., things like that. Here's an image of an estuarine system where you can see a river coming in. It has a sandbar separating the river from the marine environment, and then a delta or a mouth to that. It's not unlike the Nooksack River. As the Nooksack River comes down, there's a number of bars there that kind of separate the Nooksack River from Bellingham Bay. Salt marshes are, again, extremely productive as well. They can fix a lot of carbon and have a lot of plant growth. In general, salt marshes are one of the most impacted ecosystems, and you could think about this as they exist in a really narrow plane. So much so that the plant communities in the salt marsh can really be dictated by a few centimeters of elevation. And this is a zone that's between the marine environment and the freshwater. And it's the area that's impacted by both, so sea level rise and development in the watershed, as well as a really narrow area that has been historically already developed. And so, given that's really sensitive to that, we're losing salt marshes at a high rate, and we don't have that many left. In Puget Sound, there's very few intact salt marshes. And so this tidal gradient is that salinity gradient I'm talking about, that if you're just a few centimeters high, you can be more of a freshwater tolerant plant. If you're a few centimeters lower, you get more of a saltwater tolerant plant. The image is East Coast image of salt marsh and mangroves, which obviously we don't have mangroves here, but there actually are some invasive mangroves in Southern California, and then further south there's more mangroves. They're pretty interesting because, again, they're also a very fragile system, because anything right at that interface between fresh and salt kind of gets it from both sides, and on top of that, they're areas that have been historically developed and are continuing to be developed. Mangroves are also very important root rearing habitats for juvenile fish as well, and they provide a lot of structure. So when we look at this next image, you can see these root masses. They're pretty interesting the way that they work. They have this root mass that goes down in the water and provides a three-dimensional structure for fish and other juvenile organisms to hide in. One of the big impacts on mangroves is cutting these systems down to build shrimp ponds. And so shrimp aquaculture is a big industry. One of the issues with shrimp aquaculture is they build these big shrimp aquaculture facilities, and they're really only good for about 4 years, maybe 7 years. And then they get a disease outbreak that kills all of the shrimp, and that pond is then forever barren, and they can't raise shrimp in it. So it's this continuous cycle of cutting down mangroves to build shrimp ponds farming them for a few years and going on to the next. Also, we can have cheap shrimp platters at Costco. Rocky intertidals, one of my favorite spots. So this is dominated by sessile organisms, meaning critters that are stuck where they are. Think of barnacles. You can have like little limpets, or maybe kind of in between a mobile and a sessile. Like they're technically mobile, they can move around, but they've got very small ranges. But obviously barnacles are pretty stuck where they're going. Mussels as well, things like that. But they have this hard substrate. We'll get more into this in the facilitation chapters later in the book. But the intertidal, to me, and rocky intertidal in particular, but all intertidal. When we look at the intertidal, there's really two main forces at play here. So here we see kind of the iconic picture of the rocky intertidal and a sea star. You've got some, looks like Turkish towel here, some porphyra, some fucus here, some seaweeds, some barnacles, and the big predatory Pisaster, sea star. So critters in the intertidal want to live further down on the intertidal, so they're underwater more, so they can feed more. But they don't live further down because they're at risk of predation. So their lower limit, how far they can live down on the tide line, is driven by predation predominantly. The upper end of where they can live on the tide is driven by physical stress, by desiccation. And I mentioned that those July-August lows, when it's 80 degrees out and it's low at 2 o'clock in the afternoon, in the Northwest is when critters die. That's when the intertidal melts. So the upper end is driven by physiological stress. How well can they hide in pools? How well can they seal up to prevent desiccation? As well as facilitation, meaning that if you're a limpet out here in the open and it's 80 degrees, you're going to get cooked. If you're a limpet hiding under this seaweed, this Turkish towel, you're probably going to be okay. So you can use facilitation to move further up the tide line. Again, we'll get in greater depth on that later. Sandy shores, I've actually did a lot of my doctoral work on sandy shores. They're pretty cool. They're interesting because at the surface they're shockingly sand. But they have a lot of infauna, meaning critters that live within the sediment. Mole crabs are a big one that we have. We have a lot of on the coast. It's a different species, and there's two different species in San Diego. There's these like little, almost like flea-looking things that burrow down in the sand pretty quickly. Amagretta, I want to say. I think I'm butchering that. I'm blanking on this species now. And a lot of clams as well, and that's what I studied, were a variety of clams there. There's a shockingly large amount of polychaetes, worms in there, some copepods. Hydroids are related to, they're cnidarians, they're related to jellyfish, and they actually will take hold on one of the little clams I would study. Almost all of them have little hydroids because it's the only hard part, so you'd have this little hydroid hanging off of it. And here's an image of a burrowing clam. We should look up like an image of a razor clam, because here they have an image of what is effectively a butter clam. Not necessarily doing a butter clam thing, but it's interesting how clams dig. And so this is the siphon of the clam, where it brings water in and exhales water. So it has an inhalant siphon and an exhalant siphon. So water comes in, passes over the gills where it filters oxygen and filters out plankton, and then exhales out the other side. Clams dig by, this is frequently called the foot of a clam. They make it narrow and pierce down in through the sand, and then you can see here they fatten it out, which serves as an anchor, and then they shorten the foot. They retract the muscle. So basically they drill down, widen their foot to build an anchor, and then suck down onto that. Things like razor clams on the coast of Washington can do it remarkably fast. I dig clams here all the time, and most clams are just sitting there. They are not running from you. Gooey ducks are obviously much deeper, and it's like more of a event to get one, but it's not going anywhere. It's stuck there. If you go try to dig a razor clam, they can dig down a foot or two in a matter of seconds. It's absolutely remarkable. It's actually sporting. And so in shallow ocean zones, light penetrates to the bottom and supports sessile and photosynthetic organisms. So they're making a big difference here. Shallow ocean zones is light penetrating to the benthos. And these sessile organisms support diverse community of other organisms by providing energy and physical support. Now that's the main two things that we need to think about when we want to think about structuring ecosystems: energy and physical support. And by physical support, it can be not necessarily like support like pillars of a house, but providing a three-dimensional structure. One analogy I use a lot is, you can imagine a sand flat, and you're diving over that sand flat, swimming around. You'll see some stuff, some flatfish, little schools of, small pelagic baitfishy kind of stuff. And then you stumble across a coral reef, and all of a sudden there's this amazing diversity of invertebrates, vertebrates, and perhaps some algae as well. That's all driven by that three-dimensional space of the coral reef itself. It has little nooks and crannies for critters to hide in. So when you add three-dimensional structure to a two-dimensional space, generally you have an increase in biodiversity and biomass. You can, again, go back to the analogy of a rainforest compared to a field, the same sort of idea. Three-dimensional structure and then photosynthetic capacity driving, providing energy for the system. So coral reefs, again, are an example of that. They're restricted to warm, shallow waters. Corals form large colonies and have associated algal partners, symbiotic mutualism. So corals are interesting because they are animals, but they have a symbiotic zooxanthellae that photosynthesizes, and so they can both filter feed and consume things like copepods swimming by, or they can harness the zooplankton, use energy from the sun to fix carbon. And so what the corals are doing is they're giving the zooxanthellae a nice place to live inside of a coral, and in exchange the zooxanthellaes cutting them in on the deal and giving them a little bit of the carbon they're fixing. So many corals extract calcium carbonate from the seawater to build their skeleton-like structure. They form large reefs. This is, one of the concerns around ocean acidification, is impacting their ability to calcify. The bigger issues, typically things like either loss of structural diversity in the ecosystem, meaning there's been a loss of the ecosystem balance, and things that go by and actually eat the corals or graze on the corals have come out of balance and can overgraze the corals, as well as direct temperature impacts. When coral get too warm, they release the zooxanthellae, they get stressed and kick them out. That's called coral bleaching, which is why that's effectively the bleaching part of that statement. And then a big impact's nutrients, both impacting the water clarity, so light not penetrating into it, but also with the addition of large amounts of nutrients, you get algal growth on the coral itself, which will shade the coral out and ultimately kill it. Here's an image of a coral reef from space. This is Long Island in the Bahamas. There are many coral reef systems around the globe. None of them are doing that well. And so I mentioned a few of these. Nutrients is a big one, as well as warming that can cause loss of zooxanthellae, resulting in coral bleaching. Seagrasses are something that I really like, and I want to make a few clarifying points that I hope will serve you well. There are seagrasses and seaweeds. They are defined differently. A seagrass in this part of the world, our main seagrasses is Zoster marina and Zoster japonica, which we'll talk about in a minute. Eelgrass. Seagrasses are typically they're angiosperms. They're flowering plants. They have seeds and they have flowers, and they reproduce by rhizomes and they reproduce by seeds. Algae are not. They are algae, not vascular plants. And they're very important here in the South Sea and globally. Really sensitive to a few things. One is shading, so increased turbidity in the water, decreased water clarity. Drew Harvell just gave a good talk in the Huxley seminar series about eelgrass wasting diseases. And so eelgrass are being impacted by these diseases. I'll give that eelgrass in the next picture here. These diseases that cause lesions and ultimately death in the eelgrass. Additionally, hydrogen sulfide can build up in the sediments that will kill eelgrass. Hydrogen sulfide is a byproduct of anoxic respiration, and that typically happens in areas where you have a lot of organic matter laid down, fine sediments with a lot of organic matter. And that organic matter decays, consumes the oxygen, then anaerobic bacteria come into play and can generate hydrogen sulfide, which is a toxic gas, and has been shown to cause mortality, particularly with young eelgrass. And so you'll have these eelgrass beds that are aging out. Basically, they have a few old individuals left, but none of the youngsters have survived. Here's a eelgrass bed from Bainbridge Island, taken at a moderate low tide, but you can see it here. There's a bunch at Marine Park. We actually are just north of the largest eelgrass bed in the western coast, on the west coast of the U.S., which is Padilla Bay. So the Padilla Bay Natural Estuarine Reserve. It's a massive, massive bed. If you go there on low tide, you can't even see the water. It's just eelgrass as far as you can see. I've done a workout there, and it's insane. Just to, you'll be walking forever, and you'll never get to the edge of the water. And that's typically not the goal. There's just so much eelgrass. We also have another species. So I mentioned Zoster marina. Zoster marina is the native Pacific eelgrass. There's another species called Zostra japonica. Zostra japonica arrived here as a hitchhiker with aquaculture species, oysters, and manila clams. I'm not sure on the date. Probably early. It kind of sounds kind of when I say early 1900s. But likely around then. Pacific oysters were brought over as early as the 1800s, and they had hitchhikers on them as well. Invasive species came along, but I don't think Zostra japonica took hold until it could have been the 40s or 50s, and it could have been prior to that. What's really common, the state of Washington considers it a noxious weed, which means that you can spread herbicides on it. That's driven by the oyster aquaculture industry in Willapa Bay that wants to spread herbicides on Zostra japonica so it doesn't interfere with their oyster operations. It's typically not a big competitor for Zoster marina, the native eelgrass, because they live at different tidal heights. The Zostra japonica lives higher on the tide line. So if you were to walk from the beach, from the parking lot out, the first eelgrass you run into, particularly in the spring, is likely Zostra japonica. And then there will even be a bare spot in the mud, and then Zostra marina, the native eelgrass. Kelp forest. Now we're talking about seaweeds. These are algae, brown algae, and there's other types of kelp is kind of a broad term, but there are other types of kelps as well. And so kelp beds or forests are extremely important. They can fix a massive amount of carbon. They provide a massive amount of three-dimensional structure from the benthos all the way up to the top. Sometimes these kelps can be 100 feet tall, and so particularly in clear water areas, you can be in 100 plus feet of water and have a kelp go from the bottom of the ocean all the way up to the surface. The bottom, they have what's called a holdfast. It's a root-like structure. They're not roots though, that they use to grow around and in and under large rocks or boulders, and that's their holdfast. That's what holds them or anchors them to the bottom. And then they've got stems and fronds. We'll look at an image next here. This is Macrocystis. What's the species? Macrocystis gigantica. Yeah, gigantica. I'm pretty sure. Yeah, Macrocystis. A common name I've heard, giant kelp or California kelp. Mostly what we have Macrocystis here. We mostly have Nerocystis, which is the bulb kelp, and so it's got one big bulb and one blade. Here what we have is a stipe with multiple bulbs and blades coming off of it. You can see kind of in the back here a bit more. They're really, really pretty. And they can grow on the order of 1 or 2, maybe even 3 feet a day. So they produce massive amounts of carbon. And as you can see here, these blades are have been grazed. They've got holes in them. There's a number of bulbs missing their blades, meaning that the carbon they're fixing is being consumed right away. There's a kelp isopod. There's a number of crustaceans that actually will feed on the kelp itself or the biofilm around the kelp. And you have things like these are half moons that will go around and pick off the invertebrates from there. And so the pelagic zone, this has all been near shore stuff. The pelagic zone is offshore. It's beyond the continental shelf, where the photic zone supports the largest density of organisms within the pelagic zone. And below the photic zone, energy is supplied by falling detritus. In the pelagic zone, we have nekton, swimming organisms capable of overcoming ocean currents. Fish, sea mammals, marine mammals rather, sea turtles, squid, octopus, etc. As compared to plankton, of both phyto and zooplankton, which are at the mercy of the currents. So the definition of a nekton is somebody strong enough to swim against the current. Here's some images of marine phytoplankton and zooplankton. They're really quite beautiful. There's a lot of really cool phytoplankton out there that look quite amazing, akin to snowflakes. Over in the zooplankton, you've got I'm blanking on, it's a type of shrimp. This is actually a crab larvae, a zoea. Dungeness crab larvae look crazy. They look nothing like crab. They actually have a huge spike rostrum going out their nose and another one going across their back. So they're small little zooplankton that are fat and tasty, but they have these huge spines going out both ends to keep them from being predated upon. And then our friends, the copepods over here as well. So pelagic organisms must have ways to prevent them sinking, swimming, or buoyancy. Their body shapes or projections can help slow the sinking. So we see that a lot in phytoplankton. They'll have spicules everywhere. And those spicules actually do a number of things. One, it helps with, so like a little spiky ball. It helps with sinking, makes them larger so they're harder to predate upon. And they can also host interesting bacterial communities within those spicules that can form a mutualistic relationship as well. There's the seaweed sargassum, which has these huge gas-filled bladders to keep it afloat. So these floating islands of seaweed out in the middle of the ocean called sargassum. I've never seen it. I've always wanted to see sargassum. And below the photic zone, temperature drops and pressure increases. And so Pressure increases every 33 feet, increases 1 atmosphere, which means the pressure we feel here at sea level, if you go down to 33 feet, it doubles. You're at 2 atmospheres. If you go down to 66 feet, you're at 3 atmospheres. If you go down to 99, you're at 4. And so as you get further and further down, it's really crushing depths. And that's an issue for organisms that would typically have a swim bladder. It's hard to maintain an air bladder down there. Additionally, think about marine mammals diving all the way down there, what that's doing to their lungs. Crustaceans and copepods graze on the rain following detritus from the photic zone, and crustaceans, cephalopods, fishes are predators of the deep. This is a deep sea anglerfish, and so these are pretty cool. They've got they live down in the deep ocean in the deep pelagic zone, and they've got this light that they flicker, and then there's no light there, so critters are coming by trying to figure out what it is to eat it, and then they gape and suck and pull on their predator or pull on their prey. And similar to the land, there's been impacts really all over the ocean. So again, this is a heat map showing a variety of impact, from red to high impact to low to very low impact. And so some areas around the Arctic, you could claim that there's a low impact. Obviously, with the Antarctica melting on top of, if you think about the other issues associated with whaling or the loss of large marine mammals, there's some pretty big impacts in Antarctica as well. So that's it for the ocean. We're going to dive deeper into like all these ecosystems, and so the point is just to have a familiarity with kind of what goes where. So we're really just trying to lay the foundations of the puzzle right now, and then we'll get into the interesting biology and ecology of these systems a bit later. So that will wrap it up for chapter 3.